This is the part 2 of the vaccines. So third vaccine is basically subunit. What is subunit? Subunit is basically the conjugated molecules. As you guys all know, we are having the different conjugated molecules. Conjugated, which means the molecules which is comprised of like the two polysaccharide, okay, that is called the conjugated molecules. Okay, so how we are making the vaccines of conjugated molecules and how they are used. For example, we are getting the toxic part of what? Toxic part of the antigen, the foreign particle, okay, which is in our body. We are going to make the vaccine of that antigen. So we will take what? We will take the antigen, toxic part, any specific toxic part which is causing a disease, and that will be put into the vaccine. Okay, so we will put into the vaccine, and then we will what the toxic we have make a toxic part vaccine that we will that insert into the body. Okay, so this is also the, you can say, the subunit vaccine. Okay, if we talk about the drawback of that, so we are having the drawback like if antibody, like we are making the antibody, right, from which will kill the antigen. Okay, so drawback, if we talk about, so drawback we are having that, for example, if anybody is having the weak immune system, so that can cause like disease too. Okay, because we are doing what? We are doing the we are like inserting a you can say the toxic part of antigen, so that can cause a disease too in anybody if anybody is having a weak immune system. Okay, so we will do what? We will take you can say that the toxic part of an antigen, okay, which is causing a disease, and that we will insert into the put into the vaccine, and that the vaccine we will insert to a body. Okay, and our body will do after when we insert the vaccine. So just because of that vaccine, our body will do what? Our body will make a antibodies of that antigen to kill that antigen. For example, we are having diseases like hepatitis B. This is a disease, okay, which is occurred by the you can say the placenta, not the placenta but the breast of a woman, and you can say that this is basically the oral, you can say that the disease is there. So that is the hepatitis B, and we are having the vaccine of this, like this method, so that is successful. We are having influenza too. Okay, that is also successful, but in some cases. Okay, so this was about the vaccine. Fourth one we are having, that is the DNA recombinant. DNA recombinant, like um this is used in the genetic engineering. Okay, we are doing what? We are cutting and introducing a new gene. How? We will take a part of gene. Okay, we will take a part of gene of what? Gene of pathogen. We will take a, we will extract a gene from that pathogen which is causing a disease. So that, which is like the gene that is multiply in our body, that is multiplying in our body. Okay, that is causing a disease. We will take that gene. Okay, and we will do what? That we will insert into the plasmid DNA, plasmid DNA of bacteria. We will insert into that. What is plasmid DNA? This is the extra DNA, okay, which is used to insert into the bodies. So we will do what? Just because of that, we will insert into the plasmid of bacteria, and the you can say that the bacteria culture will occur. What is bacteria culture? Which means the growth will be occur of that. Okay, so the screening of the plasmid will occur, and the, you can say that, that is basically introduced at the commercial level, and the vaccine will be get occur just because of that plasmid. Okay, for example, we are having insulin, we are having the insulin vaccines too. It is advanced level, but it is not like affordable, it is not affordable. very like for the poor people it is not affordable. So from that you can easily, of course, it is the you can say that happen in the genetic engineering. Okay, so this is about the vaccine, which this is the part 2 of the vaccine. In the previous video we discussed about the like the active vaccine, or the like active vaccine like we were inserting a live active pathogen in our body. Okay, and one was the inactive vaccine that we have discussed in the previous video. Okay, so what, just because of these both we can easily make the vaccines. Okay, so as you guys all know that bacteria can multiply like 10 to 15 times. Okay, so just because of that we are using the plasmid of that bacteria. Plasmid is what? Plasmid is basically the circular DNA of the bacteria. Okay, so this was about.